Hi, everyone, and welcome to today's event. This is IndieBio's little corner on the internet, and we're happy to have you here as part of our audience today. We're going to learn about all of the ways that fungal biology and mycology can be used as the basis, the technological basis for a biotech startup. And I'm very happy to introduce today's panelists, Mauricio Braya, Christopher Kong, Kevin Chen, and Rachel Windsor. And so I will start with Mauricio Braya. Hi, Mauricio. Thank you so much for joining us today. Can you tell us a little bit about your role at Microma?
Hi, Julie. Hi, everyone. Thank you for inviting me here. It's a pleasure to be in this amazing panel. So I'm Mauricio. I'm the CSO and co-founder of Microma, an Argentinian biotech company developing novel natural ingredients, mainly for the food and cosmetic industry. Our technology is based on submerged fermentation of proprietary strains of Filamentous Fungi. I am a biotechnologist. I have a PhD in life sciences and a strong background in microbial fermentation. Before Microma, I worked in a biotech company developing fermentation processes to produce enzymes for the food industry using engineered strains of E. coli, other bacteria, and yeast. But then I moved to academia and got interested in bioeconomy and circular economy. And so I started researching on how to add value to agri-industrial wastes. Here in Argentina, agriculture is one of the most important economic sectors, and there are a lot of agri-industrial activities derived from that produce tons of lignocellulosic waste that are a matter of environmental concern. When I was in academia, I started working with filamentous fungi, and I worked with the fungi that produce a color metabolite, and I thought there's something here. So, and 1 year later, Microma was born, and today our main project is a red fungal colorant aimed to replace petroleum-based red colorants used nowadays in the industry that are so harmful for the environment and human health.
Very interesting. Thank you. And I'm sorry that I interrupted. I think that was my internet connection, not yours. Great. Okay. So thank you so much for joining us. I would next like to introduce Rachel Windsor, who is the lead scientist, senior scientist lead processing. Did I get those words together correctly?
It's fine. I'm the lead biologist for process development.
Process development, thank you, at Microworks. Yes, thank you. Rachel, could you talk a little bit about your background and what you're doing now at Microworks?
Sure, of course. Thanks. Thank you also for inviting me here today. I'm so excited to be here with all of you guys. My background is actually kind of broad and diverse. My undergraduate training was in protein engineering and a bit of rainforest biology. My PhD work was actually straight up mycology. I was in a forest pathology and mycology lab looking at invasive fungal and fungus-like pathogens in less managed forest ecosystems. So using molecular ecology and evolution methods to identify where invasive pathogens might be coming from and how they might interact in situ. So after that PhD experience, I did move into more of the biotech world. So I was actually at a company in IndieBio in 2016 called BioNascent that was bioengineering human milk proteins to try to improve infant formulas. I was it was my friend's company, and I was working there, and I saw this other company called Microworks there that I really, really admired, also there in 2016. But I moved on to do a couple more biotech sector things and really found myself interested in the fungal aspects of my other work, where we were trying to produce molecules from different fungi, especially filamentous fungi, and was looking for a way to bring my mycological training and my knowledge about specifically filamentous fungi and wood decay fungi back into my career. So I was so excited to find Microworks, and I've been here for about a year. So Microworks is a company that is a biomaterials company. We're using the vegetative mycelium cells of fungus to create our first launch product, which is called Reishi-Fine Mycelium. And that's can be used in leather-like applications. So like Mauricio mentioned, we're very interested in the circular bioeconomy. So taking lignocellulosic waste from agriculture and silviculture and creating something that can be used for its lifetime and then hopefully will biodegrade and return resources back into the system. So I'm sure we can talk a lot more about that in a little bit, but that's what I'm doing. So right now, like you said, in the biologist for process development, so working on the fermentation side of our process, everything from inoculating the fungus into our substrate to growing a mat of mycelium that can then be processed to become rechefied mycelium.
Oh, very interesting. Yes, I certainly will have questions for everyone here. But one thing I would like to point out, if you don't mind me saying so, Rachel, is that if you look behind her, she's actually working at home with a lab that was set up. And I just think that is so cool that Microworks did this for you to allow you to continue developing your products. Really, really cool. Thank you.
Yeah, they've been incredibly supportive and fantastic.
That's really, really neat. All right, I'd next like to introduce Kevin Chen, CEO and co-founder of Hyacinth Bio. Thank you for joining us today, Kevin. Can you talk a little bit about your scientific background and also what you do now at Hyacinth?
Yeah, definitely. Hi, everyone. Great to be here and calling in from Montreal, Canada. So my scientific background is in biochemistry, mostly focusing on structural biology and kind of drug development stuff. But I also did a few years of the iGEM competition. So I would say to some extent, I care a lot about being able to engineer microorganisms of all kinds to produce all kinds of different products. But at Hyacinth, we work principally with yeast and engineering yeast that will produce the active compounds of cannabis. So I guess yeast is a fungi, so I guess that's why I belong here. But I'll extend that by saying actually our yeast does use enzymes that are coming from originally from bacteria or from other fungi, or even from, I guess one of the key ones is coming from a protist that we're using. And so I guess we kind of like take a yeast from like fungal world and then some bacteria enzymes, and then we use that to make a plant product. So I guess we can be on any panel for any kind of organism in the end or something like that. But yeah, we're all about cannabinoid biosynthesis, and the reason that is like the cannabinoids have been shown to be like really important products in medicine and also in health and wellness, namely in treating epilepsy, where you've seen CBD or cannabidiol in particular help kids who would normally have 300 seizures per week go down to basically seizure free. So we believe in making that available to as many people as possible. And right now, growing cannabis is essentially cost prohibitive, or there's a lot of challenges in growing that plant at large scales with the right quality controls, and also making that available in any country around the world. And so we like to use a yeast system to do this because it's industrial fermentation. There's industrial fermenters anywhere in the world, and they're already being used today to produce all kinds of food and pharmaceutical ingredients. So our goal is to first of all, create these yeast strains and then make those available around the world to actually produce large quantities of cannabinoids and expand that realm. In terms of my role is I'm CEO here, so I do mostly the finance and business development work. But of course, I'm a big fan of the science, and that's about it.
Really wonderful. Thank you so much, Kevin. And finally, our panelist in the latest hour of his day is Christopher Kong, who is joining us all the way from the UK. Thank you, Christopher, for joining us. And can you talk a little bit about your background and your role now at Better Nature Foods?
Absolutely. No, thank you so much for having me on this panel, Julie. I guess quite similar to Kevin, the co-founder and head of business development of Better Nature. So why do I say so quite similar is that I basically handle all the biz dev and finance side of things, but also have a passion for the science. So how did I find myself at Better Nature? I did my master's in biochemistry at the University of Oxford. It was an integrated master's program, so it was 4 years. I did my master's on something completely unrelated to what I'm currently doing. I actually did it on the pathology of the African sleeping sickness. So looking at sort of protein-protein interactions, a little bit of structural biology stuff. But I guess how I found myself on this journey is that I realized that the food system was broken. The food system doesn't serve any of the stakeholders that it should. So it doesn't serve the human, it doesn't serve the end consumer very well. We all know that malnutrition, whether it's protein energy malnutrition or just malnutrition in general, and all spectrums, whether it's obesity or the lack of nutrients, is rampant across the planet. So that's obviously quite bad. And the leading cause of death on the planet is not COVID, as some people may think, but it's chronic health diseases like coronary heart disease, and the like, primarily driven by dietary choices, then we also know that the food system is one of the greatest contributors towards carbon dioxide emissions, and one of the greatest drivers of the climate crisis that we're in, not just in the production of carbon dioxide, but also other greenhouse gases, as well as biodiversity collapse through the extensive use of land that we're running out of. And obviously, that's largely driven by the meat industry, or the overconsumption of meat. And that is not great for the animals involved in the process. So what we do at Better Nature is that, simply put, we make protein without compromise. So protein that is delicious, nutritious, and sustainable. And we do that by leveraging an ancient technology, a fungal fermentation technology called tempeh fermentation. I say it's ancient because it originated in Indonesia 300 years ago, and tempeh is still today widely consumed in Indonesia. But it's failed to, I guess, gain mainstream appeal because of, I guess, the limited creativity that people have around the use of tempeh fermentation. So what we're doing at Better Nature is that we're the world's first food tech company focused on the process. And by using our proprietary blends of starter cultures and our proprietary blends of substrates, we're creating meat alternatives that look, cook, and taste just like meat using this fermentation process. Yeah, so the organisms that we typically use, come from the genus of Rhizopus, so Rhizopus oligosporus or Rhizopus oryzae, and we co-inoculate with different strains of yeasts and bacteria.
Oh, it sounds so interesting. Thank you, Christopher. Really great summaries, everyone. And so I'm going to dive right into some of the questions that we've had submitted, but also I think something that everybody is probably wondering is why fungi? Right, the sectors in which we have represented here, we have food, we have maybe food additives, we have materials, we have recombinant technologies. All of these are so diverse and have competing technologies. One could think right away, bacteria is one option. SCOBY is a different type of material, right? And also can be used to make different alternative food sources. So I guess what I'd like to hear is why you were convinced, either through customer discovery or through technological insight, that fungi were the way to go. And I'm going to try and leave this open. I know it's a little hard on Zoom for people to just kind of jump in. We'll see if we can read each other's body language. Worst case scenario, I will start calling on people. But I want this to be as not like school as possible, right? So any thoughts that you might have around technological or business insights into why fungi were the key?
I mean, I can jump right in. So quite interesting, actually. This slight bit of a tangent, but today was actually the Good Food Institute's fermentation symposium, which is this sort of all-day symposium about different fermentation technologies, not just fungal fermentation, but also bacteria and yeast. And one of the main challenges, I guess, when creating meat alternative products is getting the right texture. So obviously with bacteria and yeast, unless you have some sort of scaffold, it can be quite difficult. And the problem then becomes, what scaffold do you use? How does it that you create and emulate that meaty bite? If you use pure plant-based sources, so like the likes of Impossible or Beyond, you run into all sorts of different problems, primarily around the ingredient list. They have to use 20 plus ingredients in order to mimic that texture, because what they do is that they create they extract these proteins from these perfectly fine plant-based sources and have to reconstitute texture from that. I think a really cool thing about fungal fermentation is that you inherently get that texture from that fermentation process. So you bypass a lot of the issues with trying to emulate or recreate that structure post-fermentation or post-process. Especially with tempeh fermentation, because we use the full bean, whether it's soybeans or chickpeas or quinoa or whatever, lentils, because we use the full plant-based substrate, we can leverage that substrate to give us that texture without having to reconstitute it down the line, and therefore end up with a much cleaner product, a much cheaper product, and a product that still performs just as well.
Well, that's really interesting, Christopher. I would have bet when I asked that question and you answered it, that you would have said something about the umami flavor and not the texture that is associated with mushrooms and different fungi. So that's really interesting, and it actually ties what you're doing much more closely to Mycologists and the texture I imagine that you're trying to achieve using the fungi that you're working with. Rachel, can you talk a little bit about how you've been able to determine that fungi would make a good substitute for leather, and whether there were other types of materials that you considered trying to emulate?
Yeah, that's a really great question. So I'm actually not part of the Microworks origin story. Our co-founders were Phil Ross and Sophia Wang. They were the folks who were at IndieBio in 2016. And their interest in fungi actually sprang from their deep rootedness in the practice of art. So both of them were not from a biotech or science background. Neither of them focused on that. Phil has been interacting with fungi for decades as part of his art practice. So he was using fungi to create different sculptural biomaterials and usable biomaterials for years, and was really drawn to the interesting properties that you can get when you grow up a dynamic, adaptive, and stochastic mycelial network in different ways. So before it was even Mycologists, they had explored building materials. They were finding really unusual strength properties, and because of this deep exploration of fungi and what they can do and how you can use them and how they'll surprise you with how they interact, they came up with in 2016 this idea to make a leather-like material that's then has developed as Mycologists has grown into this fine mycelium material.
And when you came on board, were all of the structural components, the different species and or stages at which you might inoculate them, was that already determined? What have you, I guess, how have you contributed to building out this reishi material that you described?
Yeah, that's a really great question. We are fortunate in that we're working with some really amazing customers who can share with us properties that they desire. So one of my main goals day to day is to figure out how to guide and steer our fermentation process so that the ultimate outcome of this mycelial structure fits with what folks want. They might want something that's very, very strong and tough for some applications. They might want something that's very soft and flexible and buttery for other applications. So that's part of how we're using the filamentous structure of the mycelium to grow materials that can respond in different ways and can act in different ways when they're done with the processing stages.
Oh, that's very interesting. So almost a backwards design model where you know what the customer wants and you know that the fungi can do it, and you just have to figure out how to get them to that desired tensile strength or whatever it is about the material that you're looking for. Is that an accurate summary?
Yeah, that's right. And we've been able to actually release data on the performance of our material with relationship to traditional bovine leathers as well, and seeing that on several properties that we're looking at, like tear strength and tensile strength, and different kinds of flexibility and cracking measures and color fastness measures that we can perform in a way that that can really meet or in some cases exceed that as well.
Oh, super interesting. Thank you.
Yeah.
Now, Mauricio, when you said that you started, you co-founded Mycroma. I'm going to start getting all the mycos mixed up here. Mycroma. You mentioned that you had made a discovery of a filamentous fungi that had an interesting property. So in my mind, it seems like the technical insight was there, and then from there you built out your business model. Can you talk about whether or not my assumption is true and what that was like?
Yeah, that's exactly what happened. First, I discovered this fungi that can produce a color metabolite, and since I've been vegetarian for over 10 years, and I got used to reading food labels and reading about ingredients and learning about ingredients, I already knew about the problem that food colorants, the problems about food colorants. So, but in our case, so right now we are not working with that strain anymore, and we are developing our own strains. And in our case, our technology is quite different from Christopher and Rachel, because we use filamentous fungi as biofactories. We are not dealing with texture or things like that. we grow our strains in bioreactors. So the process is a little bit different, or it's a submerged fermentation instead of a solid state fermentation. And we decided to go to use filamentous fungi instead of yeast that could be the obvious cell host, because we think that filamentous fungi has natural features that make them great for submerged fermentation. And in the next years, we think that filamentous fungi is going to become the biofactories to produce a lot of ingredients and a lot of molecules.
So
For
did you
instance,
mention,
yeah.
oh no, please continue.
Oh, they have great secretion systems. They can naturally produce secondary metabolites, and some of them are toxic for yeast or toxic for bacteria. So there are a lot of advantages and some challenges also, but we think that filamentous fungi is the future of fermentation.
That's really interesting. The metabolites that have yeast and antibacterial properties kind of make it a very natural system in terms of not needing to add exogenous antibiotics or something along those lines, right?
Yeah. Filamentous fungi can produce a lot of compounds with biological activities, and that makes them very interesting biofactories to these molecules that can be used in the food industry, in the cosmetic industry, in the pharma industry. So penicillin is just the traditional example about what filamentous fungi can do for humans.
Correct. But lots of more applications are possible. Now, you mentioned that you switched from your original strain. Did you switch within the same species, so you had a more finely tuned, engineered strain, or did you completely change the expression factory, I guess I should say?
Yeah, we completely changed to a strain that is more suitable to grow in submerged in liquid culture media. And we are developing all the molecular biology toolbox to engineer this strain. And yeah, we completely switched the filamentous fungi.
Oh, very interesting. I'll have some follow-up questions on that, I'm pretty sure, in a minute, but I want to turn over to Kevin and say, and ask, not just say, what it was about yeast that you thought that this would be the system to use to produce your cannabinoids. And I guess my real question is, why yeast when there are other systems that could be used in order to make these large-scale production? You mentioned the infrastructure around being very similar to brewing and fermentation that you can install around the world, which I certainly believe in. But I'm wondering what your evidence was that this was the system to go.
Yeah. And I guess the broad strategy when it comes to thinking about like secondary metabolite production is to examine the metabolism of different organisms that you want to use and different yeasts or different bacterias and choose whichever one makes the most sense or has the thing that's closest to your end product. So for yeast in particular, like you do have a pretty wide selection of metabolisms that you can play with, and I would add that there's a lot of course, existing infrastructure, as you mentioned, but also the existing tools and methods to do engineering with the yeast genome as well. And that's where it sounds like Mauricio is on his way to creating molecular biology tools for his fungi, which is great. I think this is kind of the new wave, and the big difference between today's era of genetic engineering versus the 1970s when insulin was first made using bacteria, is that nowadays you can actually work with yeast, work with fungi, have super high throughput tools, use DNA synthesis to make a lot of gene edits and really play with the metabolism of these organisms. So yeast is nice because it has that ready engineerability, a lot of literature around that, a lot of tools that you can use to manipulate it, has a really great base metabolism, and for cannabinoids, this is a recombinant pathway no matter where you look in the fungal ecosystem. So we just kind of wanted something that is a good starting point, and then we built a lot of stuff on top of that with our different enzymes and so on and so forth.
And that's very useful, and it totally makes sense given that you're using genes from all throughout the kingdoms of the different world, right? You mentioned the plants and a protist even, which was very interesting. I didn't realize that that was part of the arsenal of genetic material you were using. I have a question for everyone about how to decide at the beginning or as you scale up which species is best, because that I think doing something as a prototype is one system, right? One way to show your proof that you can make the thing that you want to make. And lots of people who are on this call are probably familiar with bench scale research, but the ability to scale up and really mass produce your food additives or your leather materials or your foods that you're making, I think that's a challenge that many people face. And so I wonder if any of you could speak to how it was that you went about systematically looking at which fungi might be the best, or fungi, yeast, molds might be the best to use going forward, and whether the ability to grow quickly, so like doubling time, was taken into consideration in addition to feedstock. I know we've talked a lot about waste feedstocks, but some are easier to come by than others, and how those decisions were made on the technology side.
Okay, I can answer that question. So when we talk about scalability, first we are working with bioreactors. So in terms of the technology, there is a lot of bioreactors in the industry and available for scale up. A lot of companies that offer you can outsource the production. So in terms of the technology, is not a problem. In terms of the host cell, working with filamentous fungi is very challenging, but there is a lot of knowledge around some species like Aspergillus or Penicillium, even Fusarium species. Some filamentous fungi are used as biological, like trichoderma, and they are used in agriculture and so it's quite challenging. One of the reasons that we decided to go with filamentous fungi is, as you said, because of the alternative feedstocks that you can naturally use when dealing with filamentous fungi and designing the culture media. Even if you are working in submerged fermentation, you can use some agro-industrial wastes, and so that is something that is pretty appealing about filamentous fungi.
So you were specifically looking within the filamentous fungi, probably those that were able to deal with certain types of nutrient compositions and oxygen levels, etc. Right?
Yeah, exactly, and also, yeah, there is a lot of information about filamentous fungi in submerged fermentation, but not that much. So sometimes if you go to a very unique strain, you have to develop all the process, while you can work with some species like Aspergillus or Penicillium that you already have literature you can base on. But I think the most difficult part of working with filamentous fungus as biofactories is not the fermentation process, but the engineering the strain.
So the tools that you use in order to make the fungus do the things that you want it to do.
Yeah, exactly.
Yes, and I wonder if, well, before we get in, I do want to talk about building out tools and how that's affected not only those that are working at the single cell level organisms, but how the multicellular fermentation processes might be affected as well. But let's follow up with how you guys were able to decide which fungi to use. I mean, I would imagine for Kevin, it's a little straightforward. You want to use yeast, but you need to find what strain it is, that's going to grow again under the correct conditions that you're looking for. Chris or Christopher or Rachel, have you guys looked at various properties in order to determine not only what can do the things that you want at the bench, but how you will continue to grow exponentially as you want, of course, to have your technologies be those that are displacing conventional technologies like leather and other animal-based proteins?
Yeah, Rachel, would you like to go first, or?
No, after you.
Okay. No, absolutely. Yeah, it's very different to grow something at the bench and something you do at the industrial scale, especially with food. There's so many regulations and so many health and safety measures that need to be controlled for. So, for us, appearance is a big issue. The appearance of the end product is a big issue, and typically when fungi sporulate, they turn black, and that can be quite unappealing to most customers. So for us, finding a strain, a fungi that's able to resist that sporulation, and yet still grow quite quickly, or resist heat stress or water stress or moisture stress, was quite important. So the robustness of the fungal strain was really, really important. But a lot of our decisions were also guided, in fact, by benchtop analyses on the nutritional as well as sensory profile of the end product we wanted to create. So it's a bit of both. It's a bit of picking the right strains that can create the optimal product, and controlling for the fermentation conditions to create a product at scale, and to do it efficiently, and to do it reliably, right? So I think, going back to Mauricio's point, it's you've got the strain, but you've also got the parameters of the fermentation process that you need to control for to help scale. So it's a marriage. It really is a marriage between the two, and like all relationships, compromises need to be made. So yeah,
I like that.
I love analogy.
Yeah.
In a biotech panel.
And the processes. Yes, that's very nice. Rachel, finally to you. I wonder if, because I imagine making a mycelial mat at a smaller scale is where people began in 2016, but now as you're trying to develop this larger scale, how have you changed the parameters of the fungi that you're looking for?
Well, I just speaking more generally about the biomaterial space, because I think that your question has a lot of relevance to the biomaterial space when you're not using your fungus as a factor to make something else, but your biomass is actually what you're making. So then it becomes this really complex interplay between the material properties of your filamentous fungus, the aesthetic properties, like other folks have mentioned, the fermentation speed, trying to find the most sustainable type of biosubstrate that your fungus can grow on, trying to find sort of a biosafe fungus if you're going to have not a product that is purified from the fungus, but you're actually going to have people in physical contact with the fungus. You want to make sure that that biosafety is part of this. And then like other folks have mentioned, when you're thinking about scaling up biomaterials, yes, then you want to make sure that you can have a filamentous fungus that can behave in a more tunable and predictable way as you're scaling up, because again, when you're working with a dynamic mycelial network, there's a lot of variability, rightly so, because it's an adaptive network that's responding to its environment and can communicate, between cells in the network. So we have been focusing on one fungus so far, but those are things that we definitely think about, and that other folks in the really kind of expanding fungal biomaterial space, I'm sure, also have to balance when they're trying to create products that are the biomaterial.
Yes, absolutely. Yeah, and that's an excellent point about choosing a biosafe fungus, because there are, many are, most fungi are, but that's something to consider in actually all of the different products which are being created here. Great, so really, really great conversation. I'd like to turn a little bit more to the future of this technology and how genomic editing and the technologies around that. We've talked already about how Mauricio is building out some of those technologies, how those are applied to your systems. Has it been a big shift? I'm trying to think about when CRISPR really came online. Has CRISPR really changed the game in terms of how you guys are working with your fungi? Or are there other systems that you find are more, conventional within your particular sector, or the folks with whom you are working? Mauricio, I'm going to turn first to you because you are the one who mentioned building out tools. I wonder if you could talk a little bit about how genetic engineering of species that are not model organisms, right? These are not things that even necessarily have their genomes mapped at the or multifold level, right? So I think it would be really interesting to hear your experience around that.
Yeah, sure. Yeah, CRISPR's of course changed the way we edited cells in general, not only filamentous fungi. But
Of course.
before, engineering fungal cells was pretty difficult, very challenging. And now CRISPR opened or give us the opportunity to do it in a high throughput and faster way than before. And yeah, I think, and that's why I think that maybe filamentous fungi was not the first choice before, because they're difficult to obtain in engineer industrial strains. But now with CRISPR, we are going to see a lot more of fungal biofactories.
Yes, that's a really interesting point to see non-conventional, things that have not been the model organisms that people have used over and over again.
Yeah.
Now,
Yeah, exactly.
Yeah. Kevin, I wonder if, I'm sure CRISPR is, a work-a-day type of technology that you use, but Saccharomyces is also, I'm not sure if you're using Saccharomyces or Pitchio when we say yeast. So I should probably check that first, but homologous recombination has been long used very successfully in both of those species. So I wonder if you could talk about how you kind of balance old and new technologies as you build out your strains.
Yeah, definitely. And I can speak to maybe like so for sure, sorry, I just dropped something. For sure, the tools in gene editing for like yeast have been around for a long time, and that's one of the nice things to like have as a basis when working with these kinds of organisms. Where it gets more interesting is kind of like I guess looking at costs of like DNA synthesis and DNA sequencing that are different now than they were like even like 5 or 10 years ago, where you can start to work with libraries and more libraries of genes as opposed to individual like one gene at a time. And to some extent, new gene editing tools help with that whole workflow. But that's like one barrier where like you don't have to worry about getting the one plant gene that you know about like working in an organism. It's like you can find any gene that is kind of similar and then see if that one works instead. And then you can engineer that gene in like a million different ways and have that all actually be reasonably cost effective. It's not like a impossibility anymore. And sometimes nowadays I still see like academic papers that are kind of focusing on like, oh, we'll express this gene or purify this one protein. And then every time I see those, I'm like, well, why don't you just like, purify 100 of them? This costs basically the same, something like that.
Yeah,
now,
that's right.
nowadays almost. So there's a few things there that I find really, really interesting for kind of the up and coming and latest tools in gene editing.
Yeah, DNA synthesis, of course, and the nearly completed project. It may now be completed of SIM 2.0, right? The completely reconstructing the yeast.
Yeah, new yeast genomes.
Yeah.
This is going to be like whole genome synthesis is kind of maybe I don't know how many years we are away from just ordering a bunch of genomes instead of just one gene at a
genes,
time kind
right?
of thing.
Yeah.
Soon, hopefully. Yeah. I would like it tomorrow. Make my job easier, maybe.
Yeah, that's very interesting. We'll have to ask Jeff Buca how that project is going. Wonderful. And so I'd like to know a little bit more. I have a little bit of history with working with yeast, so I happen to have an inside knowledge, but I have no idea whether gene editing comes into play with things like food technologies and the fermentation there, or when you're trying to change some of those characteristics that we were talking about, Rachel. So Rachel and Christopher, can you guys talk also as to whether or not you were using some of these gene editing technologies as you're creating your products? And Christopher, I see you approach the camera, so I'm turning to you first.
Well, yeah, I wanted to jump in actually before Mauricio spoke to say like, we don't do any gene editing. Reason being is that, especially with food, and varies from geography to geography. I think in the US, it's still very much, I think regulations maybe are not nearly as tight, but here in the EU and in the UK, we've got novel food laws, right? Which basically means that if a food product hasn't been like developed or put on the market before 1965 or something like that, then it's considered a novel food, in which case it needs to go through a regulatory process similar to the FDA approval process to become a recognized food product that is safe for human consumption. And obviously for a startup food brand, 2 years is a lifetime. right?
Oh,
Yeah. you can live and die in 2 years. So it becomes a major bottleneck when it comes to, or barrier when it comes to perhaps applying genetic editing techniques to the development of our products. So instead, what we focus on is like basically finding the right strains, or putting them through high throughput sort of pressure selection. So like basically selecting them through evolutionary pressures rather than going into genomes and cutting them up and inserting new genes. So it does limit the scope of what we can do, but when dealing with tempeh fermentations, which is what we deal with, there's still a lot of scope when it comes to experimenting with the symbiotic relationship that certain species of fungi can have with bacteria and yeasts, and how that can affect the performance of our food products. And then beyond the choice of microorganisms, then there's also the choice of substrates, which obviously, and it was mentioned earlier in this call, sort of the fuel or the feed that you feed these bacteria, that has a massive impact on the sensory and nutritional performance of the products as well. So playing around with the genome would be very cool, very exciting, especially as a hardcore biochemist. I think that would be the ideal. But we need to work within the restraints and the constraints of the industry. So, but that doesn't stop us from, say, exploring perhaps novel ways of improving performance.
Correct. It's really interesting to consider how both the regulatory process of getting your product to market, as well as customer demand, which is, I think, in the UK and in Europe, a little more on the GMO-free side. And so that would certainly play into what it is that you're going to end up producing, because you want to have customers on the end after you've made it. Rachel, finally, I'd like to turn to you, because of course there's regulations. As you mentioned earlier, we're coming into contact with our food, but it's a slightly different relationship that customers have. People who are vegetarian sometimes still wear leather, right? So I wonder if you could talk about the relationship of genome editing with some of the things that you're using. Is it a technology that you use?
It's actually not a technology that we're using. Similarly to Christopher, we're able to do a lot of things without that, but there is in the literature some really interesting emerging gene editing technologies in the biomaterial space, where you can change filamentous fungi protein expression and do other things to get extremely different material properties. So it's a really exciting world. And as other folks have mentioned, we're just being able to start to edit these more intractable, some of these more intractable filamentous fungi. We're not even using ascomycetes, we're using basidiomycetes. So there's even a longer history of using bioengineering in ascomycetes filamentous fungi than in basidiomycetes. So folks are just starting to make some of these, to launch some of these efforts, and it is a really exciting place to watch what will happen to the biomaterial properties of filamentous fungi when you can tinker with them in that way. But as Christopher mentioned, you can also isolate novel strains. You can have strains interact with each other, and that can have really different properties. You can tune what your strain is doing by how you feed it, and you can also use kind of a directed evolutionary type approach.
Exactly, kind of the selective pressure that Christopher was mentioning.
Exactly. So fungi are just very flexible and dynamic, and will always surprise you. So there are lots of different ways to discover novel properties.
Oh, very, very interesting. Thanks. That was usually I try and know what the answer is of questions that I'm asking. Had really no idea in terms of the materials and what you guys are using. So thank you. Now I have a question that's, I think, is going to be focused a little more toward Kevin and Christopher, because it's more on the investment side. But of course, Mauricio and Rachel, if you have experience here, by all means, speak up. And that is how hard is it to find investors? How much education is needed to speak with investors as you are fundraising for the capital necessary to move your company forward? Is this a space where you can easily find someone who's like, yes, I know that fungal-based technologies are going to replace our animal-based protein? I'm sure that there are some impact investors who are probably in that space, but how much of your time is on the education side versus just like promoting, like, this is what we're going to do with our funds when you give us our money? So if you could speak a little bit about your experiences with investors in this space, that would, I think, illuminate some of the bio-curious startup founders in our audience.
Yeah, I guess I could start. It's, especially with food, you can't judge a food product, with just slide decks alone, or through waxing lyrical about how great it is. You just need to put the food in someone's mouth and ask them to tell you whether or not they like it or not. So for us, I guess it's a little bit more straightforward, to say that, hey, here's a sample. Do you like it? If you do, that's great, and then we can talk. And if you don't, then how can we improve? So when it comes to educating, investors, and not just investors, but also consumers, with dealing with perhaps niche or novel food ingredients, it all comes down to the taste and performance of the food product. And then perhaps the next question could be, okay, well then how did you get the funding in the first place to develop that prototype? Well, I think, I guess this ties in quite neatly with what we were previously discussing about how these technologies are enabled for biotech, food tech startups to start up with much lower capital requirements, right? With the reduction of costs of gene sequencing technologies, with the ease in which we're able to play with genomes today versus 5-10 years ago. You don't need that much investment to create an MVP, depending on, I guess, how complicated you want that MVP to be. And frankly speaking, there are enough investors out there that should have an understanding of what you're doing. Else, what you're doing is probably too niche, right? So if we're able to say, hey, we can create meat without the animal, then I think there are a lot of people out there that will understand. But I can't even think of, but basically, if you're working on a technology that has no relevance or no pertinence, then perhaps that is a little too niche. And if you're finding it difficult, if you're finding that you have to educate every single investor you're talking to, then I don't know, maybe the business model is too complex. Maybe
or
the messaging
could be changed or something, right? Yeah,
exactly. And you can, in a way, you want to find investors that get it. And there are enough investors out there in loads of different technologies that do understand the space very, very well. I mean, it is their job. So if you're finding that you have to re-explain every single bit of your technology over and over again, maybe you're looking at the wrong investors.
Correct. Yeah. Well said. And Kevin, I would imagine, you mentioned that the 1970s is when insulin was produced recombinantly, that the idea of making recombinant-based cannabinoids is not necessarily something that is so far outside the realm, as Christopher was saying, of what an investor knows. Is that accurate? I'm just positing here.
No, I mean, it's, I would say still a long ways to go before people really get it to some extent. I think for, I would say I do spend a lot of time on like education, or of course, in the end, you have to tailor your story to like match what the investor understands. So like, meat without the animal is like a perfect statement. Cannabinoids without cannabis is like a bit of a weird one, just because like, what's a cannabinoid or something like that? Like, it's funny I mean, having spent years doing this, it's like investor stuff is always a bit weird. But I guess the like through our like investors, like we have a SOSV, of course, in the early days. Then we have some angel investors and some strategic investment from the cannabis industry as well. So that's one thing, like where we were able to find investors that do understand like the impact of the technology, that do understand like the impact in terms of like consumers interested in this particular product, both from like a consumer product standpoint and from like a pharmaceutical product standpoint. And maybe to speak to some of the other scientists in the audience, I think a lot of cases, including ours, kind of have this like, capital requirement to get to your prototype stage, or maybe the message is like trying to treat a particular disease or condition. And then there's like a whole lot of science that goes behind that. And then you just kind of had to, again, like look for the investors that believe in that, like that technology story, that understand like what the end impact there, and how there is going to be like a long road between where you are today and like your R&D stage and getting to like products in like a person's hand that's actually like treating their condition. But that's and could talk for days about this stuff, but leave it at that.
Yeah, no, that's very interesting. My presumption was completely wrong, and there is still quite a bit of education in this.
Yeah, and I guess there, to some extent there always will be, I guess, is maybe the only other thing to add. I think there's, in the end where you're going to try and exist in some like frame of technology where you're not so futuristic it's unbelievable, but you're close enough in the future that's investable, and that it's also like simple enough that investors are like understanding of it.
Yeah. Right? Oh, very well said. Yeah, absolutely. Being just at the right, the timing is correct, right? And the way that you're positing your or positioning your company. Great. So I realize I've been taking up all of the questions. I do have two more questions. One of them was geared particularly to biomaterials, I think. So I'm going to ask you, Rachel, if you could speak on this. And then I'm going to just do a quick round robin and ask everyone where they think the future of fungal biology in your technology is going. So if you want to start thinking about that. But there was a question about IP, and I think that this is largely based because there are so many bioartists at home, Rachel, who are working with some of these mycelial mats and making things. And the concern is that by patenting these technologies, they will become inaccessible and actually decrease the innovation in this space. So could you maybe speak about opportunities for people to innovate even within the patent system that we're working with so that people can protect their intellectual property?
That's a really good question. I think one of the very cool things about the fungal biomaterial space is that it does not require a lot of, you don't need a big factory in order to make a piece of fungal biomaterial. So there are a lot of artists, there are a lot of DIYers, there are a lot of biohackers, community biologists who are making some really interesting fungal materials with a fungus that they might have on a rotting stump out around the corner. And I think that's really amazing. And in terms of getting people to kind of understand the material, and getting the possibilities of the material out there, I think that we're at a really, really exciting place for fungal biomaterials where the more folks who understand what you can do with mycelium and how it feels, and that it's not this horrible, repulsive, fungally rotting,
of course,
disgusting
yes,
thing,
exactly. Yeah.
is just, it's really positive. And so I think that we're in a space where a lot of people patent perhaps particular processes for interacting with it, but not the concept of making a biomaterial out of a fungus, out of this fungus or that fungus or any fungus. So I think there's a shared ecosystem where we can have community folks or very small scale designers perhaps can make some type of fungal biomaterial, and we can make one that performs in a different way, and then people can still explore this space, and particularly for these kind of leather-like materials specifically.
Okay, very interesting. It sounds like maybe the process itself is something that is unique to every individual, and as they're making those things, that's not something like as someone is developing their at-home process, that might be quite, or not at home, but at community biolab, at their small incubator space, perhaps that would be a different process than what you're using at Mycoworks, even if you're using that same natural mycelium, but that would select for something that would be accessible to everyone.
That's exactly right. Even in the space of materials that are kind of analogous to leather, the companies who are in that space are using different processes, and you can actually do this type of process at home. And I know a lot of people are trying to replicate this at home. It's not a very materials intensive process to get something out the other end, and it can be really interesting. So yes. So again, yeah, the IP is often around the specific interactive processes and processing of the materials, but not necessarily the concept of I'm going to make a flexible biomaterial out of fungal mycelium.
Oh, very interesting. All right, so I told you all that I'm going to ask you what the future is, and I'm really excited for this. We've got just about 1 minute to wrap up. I'm going to turn first to you, Mauricio, and say, what do you think the future? You've already led with a couple of things that I think could count, such as new submersive filamentous fungi, but please, where do you think that the future of fungal biology in your space is going?
Yeah, I think in general, fungal biology is going to be the future of production. You can produce biomaterials like Rachel or Christopher, or Rachel, or food products, or use them as biofactory or for remediation. It's like there is a lot of applications where you could use filamentous fungi, and I think filamentous fungi are going to be key to tackle all the challenges that humans have in the near future. Food security or changing production systems yeah, I can't imagine a place where Filamentous fungi doesn't fit in.
Oh,
Actually,
specifically filamentous fungi. That's a really interesting, strong statement. I like that.
Yeah. I think the filamentous fungi are so versatile organisms, and that they have a lot of application. You can use it in solid state fermentation, submerged fermentation, and I think that in my space, working with filamentous fungi as biofactory and CRISPR technology, helping us to develop engineered strains is going to, although there is today, filamentous fungi are used to produce enzymes and antibiotics and other molecules. I think that the spectrum is going to be broader.
Oh, very, very interesting. I look forward to seeing that. I'll move to you, Christopher, and ask where you think that fungal biology and technology is moving.
Yeah, absolutely. I mean, I'm exceptionally biased, I think, like everyone else who's speaking. And if I didn't think that fungal biology was the future of the plant-based or the alternative protein sector, then I wouldn't have started Better Nature, right? The vision is, extends more than just 2 years. It extends, the hope is to create a lasting business, right?
Of course.
But if we look at the challenges the alternative protein industry faces, the industry is really split into 3 camps. You've got, and they're dominated by 2. So you've got plant-based, which is the longest standing and the oldest subsector, sub-industry. You've got cultivated, so that's all your cell-based stuff. And then you've got kind of like a hybrid of the 2 that's kind of, it's growing, it's there, but doesn't nearly get as much attention, which is the fermented space, right? And I think fermentation can really be seen as the marriage between, I go on a marriage, I'm like, what? I don't know why, but
the collaboration between,
the collaboration,
yeah.
Exactly, the middle ground between plant-based and cultivated, because you're leveraging biotransformative processes to create novel applications novel applications of food products. And it's that in that biotransformation, I think with plant-based, the industry will trend towards a copycat model where, what Beyond are doing is fantastic, but I don't know, in the US, like a Kroger's or whatever, Walmart can probably create a copycat product tomorrow if they wanted to, and they're already doing that. So there's a lot of consolidation in that space already happening. The cultivation industry is super exciting and I love it, but there are a lot of scalability issues, especially around scaffolding. So I think, fungal fermentation specifically is really probably the holy grail of the space, because you can protect your IP in that, and I think that's really important. So it's very difficult to commoditize that sort of technology, and you do away with the whole structural issue, yet still create products that taste and look great. And then do great as well for the human body. So yeah, that's my piece.
Oh, very interesting. Holy grail, I like it. That's a high bar for fungal biology. Rachel, where do you think that fungal biology and technology is going in the future?
Sure, I think it's a really, really exciting time to be in the biomaterial space, but also to think about other applications for fungal biology as we have the molecular tools to understand fungi and fungal relationships more. I mean, so in addition to these amazing biomaterials that are coming online all around, all around the globe, we can also start exploring, the contribution of mycorrhizal fungi to agriculture. There's a lot of interesting research about different microbial consortia contributing to flavor profiles and helping maybe different crops be robust against the effects of climate change. People are talking about, someone in the chat just mentioned bioremediation. I mean, now that we know that we should be looking here, there are so many applications that we're just starting to touch on. So I know you guys are talking about your own spaces, but that's sort of how I think about this, and things that we haven't even talked about today or don't have represented here today, because now we can identify fungi, see where they are and what they're doing better than we ever could before.
Oh, and we could have an entire another hour discussion on where the future is outside of our own sectors. I know we're going over just a little bit, and I'd like to thank everyone for their time. Kevin, I want to make sure to let you say where you think the future of fungal technology is going.
Yeah, I'll be quick. But I like what Rachel was saying. It's like, look around you and see how many things that weren't made by fungi, and then imagine the future where everything is made by fungi or something like that, right? But I think the other direction that I want to add to this is like, of course, sustainability being a great thing for biological synthesis of things, but getting into being able to control and manipulate biology in such a way that you can invent new things as well, is kind of one of the most things that excites me the most. Where, like, of course, cannabinoids, and we can produce like cannabidiol today, no problem, but we can also produce new kinds of cannabinoids that can't be made by nature and can't be made by chemistry, and probably that applies to all of your other sectors too, where we'll have brand new colors, brand new kinds of tempeh one day, and brand new leather-like materials that cows can't make or imagine making or whatever,
Right,
I guess,
right.
something like that.
Oh, I love that. I love that idea of just creating a future that's
Yeah.
yet unknown using fungi. All right. Well, we are unfortunately out of time. I'd like to thank you so much. This has been a lot of fun for me. I've really enjoyed the conversation, and I hope you guys did too. Thank you, Rachel. Just let you all say thank you real quick. Thank you, Kevin.
Yes, thanks so much.
Thank you very much, Christopher.
Thank you.
And thank you, Mauricio.
Thank you.
And of course, thank you to Alex Hall-Daniels sitting in the background, running all the AV. And thank you to our audience. We will be sending you a recording of this in the near future, so keep an eye out. All right, I'm Julia Wolf with IndieBio. Bye all, thank you.